My name is Martin Schlumpberger. I have been in QIAGEN working on product development for almost 19 years. And among the products that with my team I've been responsible for developing is the RNeasy FFPE kit, RNA isolation on the QIAsymphony, and I've also been involved with the AllPrep DNA RNA FFPE kit. The topic of this webinar is isolation of DNA and RNA from FFPE tissue, key considerations, important aspects, and also how to isolate both analytes from the same tissue section. Before I start with the actual webinar, I have to point out that the products that are shown here or mentioned are intended for molecular biology applications and not for actual diagnosis, treatment, or prevention of disease. For successful isolation of nucleic acids from FFPE samples, there are three essential steps that need to be included. And the first one is deparaffinization to actually make the tissue accessible for further treatment with various aqueous solutions, enzymes, and such. And for deparaffinization, traditionally hydrophobic solvents such as xylene or limonene have been used. And more recently, QIAGEN has developed a deparaffinization solution that makes the whole process more efficient and faster. For example, you don't need to dry your tissue sections after deparaffinization anymore. The second step is to solubilize the nucleic acids, and the best way to do that is usually a digestion of cross-linked proteins using proteinase K or other proteinases. And what is important to realize here is that the proteinases usually digest especially also cross-linked proteins, not down to individual amino acids, but rather leave behind short oligopeptides that then at one point are still cross-linked to the nucleic acid and thereby prevent enzymatic conversion like cDNA synthesis or PCR. The experience is that DNA actually requires a much longer proteinase K treatment or proteinase treatment in general than RNA. And the possible explanation for that is the tight association of DNA with histones. So there is probably a lot more cross-linked protein than in case of RNA. And then the third steps, because we still have the protein residues cross-linked to the nucleic acid after the proteinase treatment, we need some way of removing those or getting rid of as much or as many of those as possible to make the nucleic acid a good substrate for enzymatic reactions. And this is usually done by heat treatment in optimized buffers that helps chemically breaking these crosslinks. And for that step also, there is a difference between DNA and RNA in that DNA requires a harsher treatment than RNA, and at the same time the DNA can also tolerate a harsher treatment, whereas RNA you have to be more careful that if the temperature is too high or the treatment too long, it may cause additional fragmentation of the nucleic acid. I will now go through each of these important steps and provide some data to illustrate important aspects and what we consider best practice, starting with deparaffinization. So there are certain protocols where deparaffinization is done by simple melting of the paraffin in lysis buffer. And here in this experiment, this is compared to deparaffinization using our deparaffinization solution. This is done here for DNA extraction, and DNA yield has been determined by UV vis-based quantitation like a nanodrop, and also using a fluorescent dye-based assay. What you can see immediately is that UV vis OD measurement tends to overestimate yields quite a bit. This is something that we are seeing systematically, and particularly in case of FFPE samples. So you need to be careful and mindful of what the methods you use and how it compares to other methods. And what you can also see that for both methods, we're getting quite a bit higher yields if we use the deparaffinization solution to really separate the paraffin efficiently compared to just melting. And we also did qPCR here for the amplicon is a short piece of ribosomal DNA from the 18S gene. And what you can see, basically the PCR results confirms the quantitation, the physical quantitation, in that we see quite a bit lower CT values when we use the deparaffinization solution. Here we are looking at solubilization of RNA from FFPE sections, and what is the actual time required for efficient solubilization of the RNA specifically. And what you can see here, so the time frame or the digestion times used here were from 15 minutes up to 24 hours of proteinase K digestion. The small picture here shows that in a lot of cases you need more than 3 hours for complete digestion of pieces of tissue. And for optimal RNA yield, you can see that 15 minutes or 24 hours, there is, in all cases, for all of these tissues, there is actually very little difference, indicating that 15 minutes is enough and you do not need full solubilization of the tissue. And the same is also confirmed when, instead of physical RNA yield determination, we are looking at qPCR. We get, there is a lot of variability here, which is typical for FFPE samples, but essentially we are getting the same CT values after 15 minutes that we get after 24 hours. On the other hand, what you can see on the right, looking at the bioanalyzer electropherograms, that after prolonged proteinase K treatment, 24 hours, you start actually seeing a negative effect on RNA integrity. So not only are 15 minutes sufficient, it's also better for your RNA quality. Based on the observation that RNA and DNA require different times to solubilize from FFPE section, scientists at QIAGEN actually developed a protocol for isolation of, or separate isolation of DNA and RNA from the same FFPE section without splitting samples or anything. And the way that works is after deparaffinization, a short proteinase K treatment is performed to solubilize the RNA, and then that first lysate is removed and the remaining tissue is treated with additional proteinase K for a longer time. So you have a second lysate that then, and then you can isolate the RNA from the first lysate and the DNA from the second lysate. And importantly, this patented method also allows to use in the ensuing steps optimal conditions for crosslink removal as well. So we can do a more gentle crosslink removal for the RNA and a much more stringent one for the DNA. So we get really optimal results for each of the two analytes. And of course, the benefit of getting both nucleic acids from the same section is a big benefit, not only because you get higher yields than by splitting the samples in half, but you also get the analytes from the exact same cell population. So if you have, for example, a tissue section that contains both normal and tumor cells, this will ensure that the portion of tumor cells that you isolate your nucleic acid from is the same, exactly same for both analytes. Here is some data showing why the crosslink removal step is so crucial for nucleic acid isolation from FFPE, in this case for RNA isolation. On the left, you can see yields determined by OD measurement for both lung and kidney tissue. And for different, first we have different times of PK treatment, 15 minutes at 55 degrees and also 30 minutes. And then for each of these two proteinase K treatment times, we also have additional heat incubation at 80 degrees for either 10 minutes or 20 minutes. And this is again in a buffer that is specifically optimized to break such crosslinks. And if you look at the yields, you can see that at 30 minutes compared to 15 minutes, you get quite a bit higher yield, especially from kidney. And when you do only the 15 minutes, you reach the same yield if you include the 80-degree incubation. On the right, you then can see how this isolated RNA performs in qPCR. And important here is we normalized the RNA input into each assay to 50 nanogram. And again, for the 15 minutes, 55 degree, without the actual 80-degree crosslink removal step, you can see if it doesn't matter if we do 15 or 30 minutes at 55 degree, the CT values are roughly the same. And when we include the crosslink removal step, then we get a dramatic improvement of the PCR results. The CT value actually drops from around 28 to less than 24. So this is a huge difference and shows why the crosslink removal is really essential here, or in general for all FFPE samples. And the same is true for DNA isolation as well. In this experiment, there are different crosslink removal times and conditions. You can see starting from 30 minutes at 90 degree up to 90 degree, 30 minutes or 1 hour, and then compared to 70 or 80 degrees for 1 hour or for 4 hours, and also compared to just a prolonged proteinase K treatment overnight at 55 or 56 degrees centigrade. And again, on the left is the DNA quantitation. Sorry, this is DNA, of course. And you can see the highest yield we get with the 1 hour at 90 degree. And with the prolonged incubation at 56 degree, we can in some cases get almost as good DNA, in other cases not, so there is a lot more variability. But what is important when you look again at the qPCR results on the right, and again here the DNA input is normalized, you can see that in this case the 56 degree incubation is not good enough, even if you do it overnight. And the best results really by far are those obtained after 1 hour at 90 degrees and pretty much the same at 4 hours for 4 hours at 80 degrees. But anything less than that, lower temperatures or shorter incubation times, lead to poorer performance in PCR. And in this case for both a long and a short amplicon, 66 and 500 base pairs. Again, from the 18S ribosomal DNA gene. Another important complication from FFPE samples is that the formaldehyde fixation and storage can result in deamination of cytosines, and this would eventually result in artificial C to T transitions that can then show up as false positives or false variant, especially when you do sequencing of these DNA molecules. And to exclude those artificial mutations from your analysis, the best way is really to do a UNG treatment, uracil N-glycosylase. This will cut out the deaminated cytosine. So deaminated cytosine is uracil, and the enzyme removes that. And so those affected DNA strands are not included in your sequencing reactions and will not give false mutations at the end. So you can see that on the left, the number of mutations that are present at very low frequencies. And you can see that the UNG treatment removes those mutations. And then those mutations that are reported at a higher frequency, the true mutations, they are maintained. And this you can see on the right. Here, these are the mutations, the true mutations, and those are not affected by the enzymatic treatment. We're now moving to quality control of the isolated nucleic acids. And for DNA, one aspect that people are looking at is the integrity or physical length of the isolated DNA. And here you can see two examples from actually QIAGEN kit used for DNA isolation. One is the QIAAMP DNA FFPE tissue kit, and the other the GeneRead DNA FFPE kit. And what you can see easily is that the DNA isolated with the GeneRead kit is a little bit shorter than with the QIAAMP kit. But so you would normally say that the DNA quality is maybe worse. But in reality, when you look at PCR performance, here again, two different length amplicons from the human prion protein gene, 78 and 465 base pair amplicon lengths respectively. You can see that actually the GeneRead, the DNA isolated using the GeneRead kit gives a better CT value, even though the DNA looks less intact on the gel. And so that is true for qPCR, that the integrity is not really a main indicator, in part because short DNA often performs better in qPCR, and also because the physical length of the DNA does not reflect to what degree the crosslinks are actually reversed. And with respect to sequencing, since we are looking at a 465 base pair amplicon here, you can also probably agree with me that this is easily long enough for all the standard NGS application or library preparation protocols. So this is not really limiting in any way. In case of RNA, integrity is usually assessed using capillary electrophoresis with instruments like the Agilent bioanalyzer or the QIAXL from QIAGEN. And those instruments often provide something like an RNA integrity number, in this case here shown again for the bioanalyzer, for a different RNA samples here. RNA was isolated from rat tissue sections either shortly after fixation and embedding. This is the T0 on the left, and also after 12 months of storage at either 4-degree room temperature or 37-degree. And what you can see immediately is that the RNA integrity is very different for all of these, but it's also different for the room temperature and 37 degrees, but the RIN numbers are actually fairly close together. So they, in our view, do not really fully represent the quality difference. Like in, for example, for the brain tissue, after 12 months at room temperature, you can still see a bit of a ribosomal peak here. And for the 37-degree sample, it's much more degraded, but the RIN number is actually not dramatically different. So in our mind, or in our view, for RIN numbers below like 4 or so, they do not provide that much information anymore or much differentiation. And apparently people at Illumina also came to the same conclusion. So they suggested an alternative parameter for RNA integrity, especially for sequencing, which they call DV200, which I will comment on in the next slide. DV200 is quite simply the portion of the fluorescent signal that comes from RNA fragments longer than 200 nucleotide versus those from shorter fragments. And the rationale behind that is obviously that RNA fragments shorter than 200 nucleotides are considered not very useful, at least for long RNA sequencing. And the purpose of such a measure is to compare different RNA samples with regard to preservation of RNA integrity and what kind of sequencing results can be expected from those. What is important to keep in mind is, first, the electrophoregram shows the physical integrity, but it does not reflect the chemical modification by formaldehyde, the crosslinks that may be remaining or remnants of the crosslink proteins that will interfere with cDNA synthesis or PCR amplification. And to include that aspect of nucleic acid quality, the best way is really to perform qPCR on these samples. And the other thing that is important to keep in mind, that this parameter is not really suitable to compare different RNA isolation methods and how well they preserve RNA integrity, because different methods and protocols often differ in how well they isolate short RNA fragments. So some methods that do not isolate any RNA below 200 nucleotides will pretty much always give high DV200, even if the sample is not great. The yield will just drop a lot. So that is important to keep in mind. To summarize that aspect, nucleic acid integrity or length distribution is important, but it also depends on the downstream application that the nucleic acid is used in. For NGS-based analysis, often you need a minimum length of maybe 200 base pair or longer, whereas for PCR and qPCR, short fragments can still be analyzed and also provide really useful information. And in many cases, when you have a kit or protocol that isolates short fragments, like you see here in the example on the right, this is a pretty old piece of data. It's actually quite easy to modify that protocol to not recover the short fragments in case they are really unwanted. And before I conclude, I just want to mention again that this webinar is part of a series of webinars about various tips and tricks on FFPE samples, what are the best or what are pre-analytical considerations, considerations for nucleic acid isolation, and also for assays that can be performed on those. And that brings me to the end of the webinar. Thank you again for your attention and your interest.
QIAGEN.
Sample to insight.